Welcome everyone to Genetic Alliance's webinar series, What About Privacy? Look for the study of bioethical issues on the report that they released, Privacy in Progress in Whole Genome Sequencing. We've had several webinars already. In fact, this is the fifth one. All of them are archived on our website, so you can look at them there if you'd like to see them. This one will also be archived once it's over. It takes us a day or two to get that up on the web. Both the presentations will be there as well as the audio portion and the questions, so you can listen to them later. On today. Tanya, if you could give me the slide with the recommendation. So the recommendation 3 addresses consent and recognizes that it's not unique to whole genome sequencing, but that the consent process should be around ethical clinical care and research, that patients and participants have to be educated about the potential risks with regard to whole genome sequencing, and that we should look at how data is stored and shared, the types of data that someone might receive or expect to receive, and then the likelihood some of the implications of these results might currently be unknown but could be discovered in the future. And that respect for persons requires obtaining fully informed consent at the outset of diagnostic testing or research. Generally, the thrust of our conversation today will be on consent. And without further ado, I'm going to introduce Amy McGuire. Amy's the Associate Professor of Medicine and Medical Ethics.
I'm going to talk first about this important recommendation regarding informed consent. And as Sharon just summarized, this is basically what the recommendation says at a very sort of high-level view, that at the outset of testing or research, the informed consent process should include information about what is whole genome sequencing, how data will be analyzed, stored, and shared, what types of results can the patient or participant expect to receive, if any, and information about future uncertainty related to the data. And so also, Sharon mentioned that there are really 3 pathways through which you can obtain whole genome sequencing: clinical care, research, and through direct-to-consumer testing. And direct-to-consumer testing companies are just now starting to move into this space of whole genome sequencing. So I want to start by just having a little conversation about why we need a recommendation regarding informed consent, because it seems like obvious that you should be getting informed consent and that you should be informing people about the risks and benefits of whole genome sequencing prior to doing it. But my experience has been that current practice is very variable with regard to the quality and type of consent that is obtained prior to engaging in this type of work. And so I've looked at many, many research consents. This isn't based on any particular study that we've done, a comprehensive study, but I've looked at many research consents in the context of serving on different governance bodies as well as IRBs. And from my experience, and I'd like to hear other people's experience, but the language about genetic analysis, sequencing, and data storage is not consistent at all when you look at different consent forms in the research context. And most legacy samples, particularly those that were collected with consent forms that were developed prior to about 2007, really do not talk at all about data sharing, which the recommendations, the President's Commission recommendations talk about. In the clinical context, I think there's also quite a bit of variability. The majority of states actually do not require specific informed consent by law to order a genetic test. And my experience has been that genetic testing is sometimes ordered by non-genetic specialists for diagnostic purposes under a general consent to treat. So they may not even be getting specific informed consent for certain types of genetic testing. I think and I hope that with the introduction of whole genome sequencing, which is increasingly more complex, that this is changing and people are actually talking about the tests that they're ordering and the benefits and risks of that. But I think there is still quite a bit of variability across different laboratories as well as clinical practices in the country. And then with regard to direct-to-consumer testing, the company consents is really dependent on how the company develops those consent forms, and I think there's also quite a bit of variability with regard to that. So I want to focus in my few minutes on sort of high-level challenges that I see with regard to obtaining informed consent, obtaining good informed consent for whole genome sequencing in these three areas. And this has to do with the length, complexity, and quality of informed consent documents and processes, understanding of what's communicated in the informed consent process, the potential for incidental findings and the scope of informed consent with regard to that, and then unexpected findings. So the first topic is, or challenge, is one that I think everybody who has engaged in research or clinical care is very well aware of, which is that informed consent documents have become very, very long, they're very complex, and they're often not very well written. And the process, which is even more important, the process of communication with the individual who you're trying to obtain consent from, can be very variable, depending on who is obtaining informed consent, their level of competence and knowledge about the tests that they're offering, and the time that they have with the individual. And so there are some model consent documents that are out there. The NHGRI has a helpful website that has some model consent language for research consents. Up here is one that we've developed in the context of a cancer biorepository that we have here in Texas. That's an annotated consent that tries to provide some more background about the language you might want to include in each section. So these are very helpful, but even these model consent forms are admittedly very long and also very complex. And as I mentioned, the consent process itself can be very variable. The second issue is actually the one that I think is the biggest challenge to obtaining informed consent in genomics, which is ensuring that the individual who you're obtaining consent from understands the risks, the benefits, and what you're asking for their authorization for. So I'm just going to give you a little bit of background kind of on some work that we've done in this area trying to assess understanding in the research context. And we actually conducted a randomized trial of different types of consent for genomic research, and we were randomizing the consent forms based on options around data sharing. We randomized 335 consent decisions, and we followed that up with interviews with participants to assess their understanding, their perspectives, and their preferences about data sharing and genomic research participation. And we interviewed 229 individuals. And in this study, what we found was that the participants that we spoke to self-reported that they felt that they understood the decision about sharing their data and participating in genomic research fairly well. So we asked them questions like, how much do you agree or disagree with these statements? I felt sure about what to choose with regard to sharing my genetic information. The decision to share my genetic information was easy for me to make, and I feel I've made an informed decision with regard to sharing my genetic information. And the vast majority of our participants felt fairly strongly that they agreed with these statements. They felt comfortable with the decision that they had made. Yet when we looked at their actual understanding and asked them some objective questions, I think it was a little less optimistic. So we asked participants, do you know if you're participating in a research study? And 40% said no. And this was after they had signed their consent to participate in this genomic study, the one that they were being enrolled into. We asked, have you ever heard of genetic studies? And 28% said no. We asked if you have already given DNA to your doctor. 16% said no, and 36.6% said they did not know. And when we asked, did you remember signing a consent form to participate in a genetic study, 26% said no. And this did not vary based on the consent type. I will mention that all three consents that we were randomizing individuals to were written based on some of the model consent language that's out there. And so I think based on current thinking, they were fairly well-written consent forms. At least they were very well thought through, and they were developed with the involvement of focus groups with participants to get at what they wanted to see in the consent form. There was some association between the study type and I think that had to do with the consent process, because the conversation, even though the consent forms were all the same, the conversation between studies that we were enrolling from was very different. And so the two studies that had sort of the most robust consent process, their participants were more likely to know that they were participating in a research study and to remember signing a consent form to participate in the study. So this may seem disturbing and surprising to a lot of people, but this level of understanding is not unique. Understanding about genomics or data sharing in particular. So there have been a whole host of other studies that have looked at research participants' understanding of their participation, and uniformly they have found that there is quite a lack of understanding risks from participation. 63% did not understand that. 70% did not understand the unproven nature of the treatment. 29% did not understand the uncertainty of benefits to themselves, and 25% did not understand that trials are done mainly to benefit future patients. Similarly, a third study, and there are many of these studies that show similar results, showed lack of understanding regarding nature and purpose of the study, study procedures, and major risks. So there have also been a couple of studies that have looked sort of more systematically at research that's tried to improve the informed consent process. And they have basically concluded that efforts to improve understanding in the informed consent have had only limited success. And it seems that the most effective intervention still seems to be one-on-one education with the participant, which of course is very time-consuming and can be difficult. So with regard to the President's Commission's recommendation regarding understanding, in recommendation 3.1, they say that researchers and clinicians should evaluate and adopt robust and workable consent processes that allow research participants, patients, and others to understand who has access to their whole genome sequences and other data, and to know how these data may be used in the future. So I think there is quite a lot of research that needs to be done to try to improve the consent process and to think about other innovative strategies for improving understanding, because it can be very difficult to accomplish. The third challenge that I wanted to raise for discussion relates to the recommendations discussion about disclosing the potential for incidental findings in the informed consent process. And Zach Rouhani and his colleagues have pointed out that there is the potential to identify and uncover many, many incidental findings when you're doing whole genome sequencing, and they think this is one of the greatest threats to genomic medicine if we don't figure out how to deal with the analysis of incidental findings and the disclosure of relevant findings to research participants, patients, and consumers. There have been several approaches that have been recommended. One of the most popular ones now, I think, is this idea from Jonathan Berg and his colleagues to categorize results according to different bins based on their clinical utility and clinical validity, and then to treat the return of results based on sort of these categories. It's proven, of course, more controversial than we might think to figure out what belongs in which particular variants or genes belong into these different categories and how to define those. But this idea of categorizing or binning results seems to have gained a lot of traction. Some of you may be aware that the American College of Medical Genetics and Genomics recently published recommendations for reporting incidental findings in clinical exome and genome sequencing. This report is really dealing with bin 1 in the Berg sort of bin schema, and it's really dealing with incidental findings that have very high clinical significance and utility and are potentially actionable. And so the recommendations from the ACMG is that the potential to identify These incidental findings, there's 57 genes that they're recommending go on sort of a minimum list of incidental findings that laboratories would be obligated to report back when they engage in clinical exome and genome sequencing. And the ACMG recommends that the potential to identify those 57 genes as incidental findings and the reporting back of them or disclosure of them be communicated during the informed consent process to patients, and that they consent to that when they consent to whole genome sequencing. I think those recommendations are probably consistent with the President's Commission recommendation on the treatment of incidental findings. They say in recommendation 3.3 that you must make individuals aware that incidental findings are likely to be discovered in the course of whole genome sequencing, and that the consent process should convey whether these findings will be communicated, the scope of communicated findings, and to whom the findings will be communicated. They also call for additional research to study and evaluate proposed frameworks for offering return of incidental findings and other research results, and to investigate the related preferences and expectations of the individuals contributing samples. So the last challenge I just want to bring up real quick, and I'm hoping we can discuss some of these in more depth during the discussion time, is the potential to discover unexpected findings. And so this is just one example, but I suspect that as the technology improves and as we start doing more of this, we will come across unexpected findings that may have medical implications, but may also have other social implications. And so this was a paper that was published in Lancet out of a group here at Baylor, where they were using SNP arrays. It wasn't whole genome sequencing, although the same result may be found on whole genome sequencing, where they were able to identify from children who had been sequenced and genotyped the identification of first-degree incest or consanguinity based just on the child's sample. And this raised really important ethical and policy issues in terms of what kind of consent were they obtaining? Was it necessary to include the potential to discover these types of findings in the informed consent process? And what were some other obligations of the researchers in most of the cases that we've had here at Baylor? The biological mother has been a child herself, a minor under the age of 18, and so it has triggered reporting requirements related to potential child abuse. So we've looked at some of those issues ourselves regarding the reporting requirements and the consent requirements for this, and the ACMG has recently come out with guidelines and standards for documenting suspected consanguinity as an incidental finding of genomic testing. So I raise that just to kind of bring awareness of that issue and other potential unexpected findings to individuals who are engaged in using this technology in these settings. So I just want to end by saying that I think informed consent is incredibly important. I think it's a cornerstone of the principle of respect for persons and how to implement that. However, I think that it can't always carry the entire burden of the ethics of conducting whole genome sequencing, and I do think we need to think more creatively about mechanisms that have been developed to try to engage participants and patients more longitudinally, more actively in decisions about whole genome sequencing. And this is a paper that was written about participant-centric initiatives to engage individuals in the research context in different ways. So I will end there and pass it to the next person.
Great. Thanks so much, Amy. And while Tanya is moving the controls over to Susan, I'm going to introduce Susan. And Amy, I always say that the informed consent cannot bear the burden, but I don't have a great horse slide like that, so I'm going to have to steal that. So next we're going to hear from Susan Wolf. Susan is the McKnight Presidential Professor of Law, Medicine, and Public Policy, and also the Fakhri, and I'm not sure how to say that even though I have friends there, Baker Daniels Professor of Law and Professor of Law and Medicine at the University of Minnesota Law School, and in the med school as well. So without further ado, Susan.
Thank you, Sharon. It's a pleasure to do this. I want to zoom in on a piece of the debate that Amy has beautifully opened up for us, which is the question of return of results and incidental findings. I'm having trouble advancing my slides. Oh, there we go. in research genomics and in clinical genomics. I want to tell you from the start some of my punchlines here. Research routinely generates incidental findings and individual research results of potential clinical or reproductive importance. They're kind of built into the research enterprise we now recognize. And dealing with those is extremely difficult because when you think about it, you're finding in the research sphere with its own rules and regulations and laws and customs information of potential significance over in the clinical sphere. That's the structure of the problem. But bioethics and health law conventionally have made a big distinction between the clinical and research spheres. So this problem ends up challenging that dichotomy and posing really interesting problems. I think we've made a lot of progress on the research side of the fence. Now we have a big debate that's opened up in part thanks to this very interesting work that the ACMG has done on clinical sequencing, and I want to talk briefly about all of that. And this is just a quick overview of our own work. We've been working on these problems for about 10 years now, and it's interesting how much traction these issues have and how much currency. It's wonderful that NIH is robustly supporting progress. Let me just pause for a second to make sure we're all on the same page. I'm going to propose some definitions here that are already in the literature, but one of the difficulties is I think some of these definitions are now being redebated as we figure out what we mean by incidental finding. In fact, Amy used an additional term, unexpected finding. So there's legitimate debate right now, but let me at least lay out what I think of as an incidental finding. This is from our first consensus paper, a finding concerning an individual research participant that has potential health or reproductive importance. It's discovered in the course of doing the research, but it's actually beyond the aims of the study, which is why this little figure is outside the rectangle signifying the stipulated aims of that study. You can contrast individual research results, which are actually on the focal variables under study. So, you know, it's what you were looking for, but this is as to a particular individual. You worry about whether, this is of such clinical significance that maybe it should actually be individually offered to this person, which is why the cartoon figure is inside the aims box. And all of that contrasts with what we usually think of as research results, which is the aggregate results that you got the funding to generate and you're going to proceed to publish, and those are about an entire research population. As Amy suggested, the Presidential Commission's report on privacy and progress in their recommendation 3 on consent do address incidental findings in this whole domain of research results, though they're doing more focused work on it now. But at least in the Privacy and Progress report, they identified the chief ethical principle governing whole genome sequencing as respect for persons, and then stipulated that that requires fully informed consent, and consent that states what data and information, if any, might be returned. They went on to say that researchers, clinicians, the ... is commercial companies, must make individuals aware that incidental findings are likely to be discovered, as Zach Ohani's work and others have demonstrated. And the consent process should convey whether these findings will be communicated or not. Should it be to the primary care physician or a specialist? Should it be directly to the participant themselves? When you have a minor or a person without decisional capacity, you get into other folks, the guardians, parents, or surrogates that might receive the information. But this report, the 2012 report on Privacy and Progress itself, really takes no position on the scope of the findings that should be offered, the process for returning those. I haven't included it in the slide, but Amy rightly points out the important recommendation that funders fund research so that we have an empirical basis for addressing the further questions. So what are those questions? Well, let me first focus on research. But let's back up for 1 second. If you look at the Presidential Commission zoom in on incidental findings and return of results in the context of Recommendation 3, there's something that I want you to notice. Look at the second bullet, researchers, comma, clinicians. Researchers and clinicians are being addressed together. And one of the interesting and I think controversial dimensions of what the Presidential Commission is doing here is they're not making a sharp distinction between what should happen in the domain of research and what should happen in the clinical domain. I want to suggest, and I'll be interested in the discussion, that that is a problem. That what is core to the debate about research, the return of research results, and the return of results in clinical is the differences between research and clinical sequencing. For example, in research, there are big questions of the analytic validity of what is discovered. Do we really even know what this variant is? Was it ascertained or confirmed in a CLIA-certified lab, as would be required in the clinical domain? What is the clinical utility of this result? It could be the person, the PI, and the entire team is really not comprised of clinical experts who are equipped to interpret, much less return the information. And a lot of genomic research, as the people on this call know, is not done in clinical trials. It's done in human subjects research that is non-clinical. So it's exactly the differences between the research and the clinical domain that make this a very difficult problem. And I think it's problematic to mush them together, if you will, because it obscures the exact issues that are really vexing people working in this domain. If I had to guess, it may be because of some emerging, very preliminary work on learning systems that the Commission chose to put the two domains together. But I think, right now we have regulations, we have law as well as ethics that appropriately direct us to some of the differences, at least the domain we're talking about in this webinar, clinical and research genomics. So I'm going to first suggest, and I'm not going to go through every bullet of every slide because of our time, but hopefully the slides will be a prompt for some questions. Here are some of the questions that really have troubled us in the domain of research. What criteria define the domain of returnable incidental findings in individual research results? What about ones beyond clinical significance, reproductive significance, or even personal significance or utility? Is there a duty that devolves on researchers to actually aggressively hunt for these things, or is it the stumble upon scenario? And once identified, how should these be evaluated? Some teams will need to consult with clinical experts. The CLIA issue has been a big one. What should then be disclosed, and to whom, and how should you plan for all of this? Here I've already suggested is that core to this problem is that in the research world, you're discovering findings, but worrying about their potential significance across the bridge, if you will, in the clinical sphere, where the ethical framework and the duties are quite different. And that's core to this problem, and probably core to a lot of translational science that is itself moving between research use and clinical use. And that is one of the most difficult problems here. How do you create an ethics and a law that can move across that bridge? This is a challenge to the traditional dichotomy that has structured both health law and bioethics. I'm not going to go through this whole slide. It's an attempt to schematize very roughly this dichotomy, but look down to the bottom. If you've got a doctor-patient relationship, by which I mean a clinician-patient relationship, but I'm just schematize the whole thing. You've got a professional with a robust duty of clinical care, duty to serve the patient's interest, patient capacity to hold that physician directly accountable. The duty is conventionally to seek generalized knowledge and scan down. What we are talking about with the return of results debate and incidental findings is what is the informational obligation, what information should be returned, and that's still up for grabs. There are recommendations out there. We've generated a couple sets of recommendations, most recently a year ago in Genetics and Medicine. One of the things that we argued is that it's important to distinguish should return from may return. We actually had a third category of do not return, and that's kind of stuck. A lot of the recommendations do this distinction between should and may. And if you look in should, analytically valid, compliance with law, where CLIA is really in the center of the target, established and substantial risk of serious health condition, actionable, and you've got consent from the participant at some point along the way for the return, for the offer. Fapsitz et al., Richard Fapsitz led a big group. A lot of us were in it, about 30-some people under the auspices of NHLBI. Again, should, may, and very similar to what you just saw, important health imports with law. But pause for a second. Are in the context all of research. So they have garnered some important objections that this represents a diversion of resources from research to really what is a clinical function, that it imposes clinical duties on researchers who may not have the proper expertise, that it invites confusion between research and clinical care, exacerbating the therapeutic misconception. In other words, those are arguments that, hey, it's research. Stay on the research side. Don't go marching into the clinical side. Nonetheless, the technology itself is moving into the clinical sphere. The ACMG Board of Directors itself a year ago recognized this, that for some primary indications, genomic sequencing is moving into that sphere. For example, cancer patients who've run out of treatment options and may be sequenced with their consent for the purposes of finding additional targets for therapeutic interventions, or kids who have perhaps a severe phenotypic disorder and gone through a lot of testing on a diagnostic odyssey, no one can figure it out, and sequencing is recommended clinically for the purposes of getting to the bottom of what's going on. Many anticipate, of course, an even more robust future of affordable sequencing. So when you look at the Berg et al. Recommendations, the bins that Amy put up, it's important to recognize that that's a recommendation in the clinical sphere for clinical whole genome sequencing. But it's interesting that, again, should, may, should not, and it looks very similar in some ways to the research recommendations, though I actually think this is a controversial point about Berg et al. Because clinician duties are much more robust. If should return is only medically actionable, and medically actionable is defined narrowly in terms of what a doc can do about it, there's really an interesting debate to be had about whether that comports with our concept of informed consent and what physicians should share with their patients in the clinical sphere, where really it's typically a more patient-centered standard. And some of what clinicians do feel bound to return goes beyond what they can fix to things that are important for the person's own personal planning. Let's zero in then on the ACMG evolution, which is a very interesting one, as Amy suggested. In 2012, the ACMG issued a policy statement on clinical whole genome, whole exome, looking at incidental findings. This is kind of their first cut on it before the more recent 2013 work. And there they recognize that incidental findings are highly likely, if not inevitable, when you embark on whole genome and whole exome sequencing. Very importantly, if you scan down several bullets, they were careful to allow patients to opt out of receiving some incidental findings. They acknowledged that individual cases might push on that, but their rule was a consent rule, that patients really ought to be able, of course, to consent, and that comports with the history of medical genetics and now medical genomics, that we don't, throw consent out the window because the technology has moved forward. 2013 was when this very interesting report, Green et al., that Amy flashed up, came out. This was just in March, recommendations for reporting of incidental findings. Again, remember, we're in the clinical sphere here, clinical sequencing. And that report specifies a minimum list of 57 additional genes that labs need to analyze, ACMG recommended, whenever they perform sequencing for another primary indication. And it's a heterogeneous list. Genes associated with specific cancers, cardiovascular conditions, malignant hyperthermia. But what's very important is that beyond the kind of global consent that the report envisioned when the patient was consenting to sequencing, a kind of warning, hey, we're going to look at additional incidental findings, there was a rejection of the idea that patients should be able to consent or not consent to these specific genes. And remember that this was postulated as a starter list, with the notion that labs could add others, and that ACMG itself would annually revisit this list, and the list might expand. So ACMG said that the labs, if the patient, remember we're talking about some very sick people, the cancer patients, the kids with the anomalies that are defying understanding and diagnosis, so people want to go to sequencing. If those people in whom sequencing is recommended want the sequencing, then they have to agree to this additional analysis, and the lab must report the results of that analysis to the clinician, and the clinician has to report this to the patient. There's an explicit rejection of the traditional right not to know. And ACMG issued a clarification in April saying that failure to report is actually unethical. So there's been a debate, and this is just a piece of the debate. People might be interested that Amy led a very interesting paper that came out in Science on May 31, and I with George Annis and Sherman Elias. It was a kind of counterpoint that Science was really, I think, thoughtful enough to feature, but there's a lot of work coming out debating pieces of this puzzle. And I think the controversy is really core to the topic we're talking about today, consent on autonomy. ACMG itself, to its great credit, in the March initial statement, was very candid. They said they did not favor offering the patient a preference as to whether or not to receive the minimum list, and they recognized that that could be seen to violate existing ethical norms on autonomy and right not to know. And there is a tension, I think, between that and the 2012 statement that patients should be able to opt out. On kids, there's another debate that's emerged because ACMG urged in March that their analysis apply and their recommendations apply regardless of patient age, despite the long-standing consensus that you don't do genetic testing in kids unless medically necessary before the child achieves majority and can decide for him or herself.
1 minute warning.
Yes. So where do we go next? Well, we've got guidelines. They're not in complete alignment, but really there's a lot of consensus on what to do in research. And consent is right at the center of that. But there's a new debate now on clinical, really prompted by the ACMG recommendations, and consent is really a big issue in that debate, as I've suggested. So let me conclude. I think this problem of return of results and incidental findings is fundamentally translational. It's about a science that's moving from research to clinical, and it's about how we move information from research to clinical. And then in the clinical sphere, it is at core, if you flash to the last bullet, it is at core about patient choice and whether we're going to hold to our centrality of patient autonomy and the necessity of patient consent, even when we move into an era of clinical genomic sequencing. Thanks very much.
Thank you. And now we're going to move to John Wilbanks, who is a, John, you're going to have to tell me your position with Sage Bionetworks and also a fellow at the Kauffman Foundation.
I'm the Chief Commons Officer at Sage, which basically means that I figure out how to share stuff as well as possible. And what I'm going to talk about is a little bit different from the previous speakers, and I get the benefit of going last. So I'm going to talk more about the experiences we've had at Sage developing some new methods for informed consent online, and rather than address the incidental findings for consent, it's been covered pretty well. So the first thing that we came to when we were trying to design an informed consent process is that the process itself needs a rethink. So this is a shot of the earliest consent form I could find on the web. It's from yellow fever experiments over 100 years ago in Cuba. And what's funny is that if you look at the English translations, the fundamental structure hasn't changed that much, despite the fact that we're talking about radically different kinds of information now. It's not just drawing a blood sample. It could be something like your sequence. It could be something digital, something from your phone, something about your longitudinal phenotype. Right, so we've got all this capacity to gather new kinds of data, including whole genome sequences, but the consent forms have remained relatively stable in their structure, and the consent processes have remained fairly stable in terms of talking to your doctor. So what we came up with were some principles for scaling consent alongside the capacity of an individual to gather their own data. So again, it's a little bit different than WGS and incidental findings, because increasingly people will simply go get their whole genome done outside of a clinical context. So the question is, how can they then do something with it at that point? And the three things that we came up with is that the controls that the consent imposes on researchers are going to be necessarily weak, because it's very difficult to scale strong controls on data once that data has been made available. It needs to be simple, or it won't be understandable either to the person going through the consent process or the researcher who's trying to use the data. And ideally, it needs to be open. And just to make sure what we mean by definitions, weak doesn't necessarily imply weakness, simple doesn't imply simplistic, and open doesn't imply free of charge or free of constraint. It's more that the process is transparent, the constraints or the costs are obvious, and that they are even and fair to everyone involved in the system. So the reason we did this is that we think that people will come to a time where they simply get their own genotype or they get their own genome sequenced, and they'll be able to do something interesting with it, but we're not really near there yet. Stephen Friend, who's the CEO at Sage, has written about Metcalfe's law and the need to have a network effect emerge out of genome sequences, but we're a long way from that. And what we're trying to do with the consent process I'll show you is to basically try to stimulate the capacity for unanticipated benefits to come out of people sharing their genotypes or their genomes. And it's not happening yet. So I actually tried mine. This is my genotype I'm showing you inside the Sage Synapse data storage system. I've gone through the consent process I'll show you in a second, but I wanted to show you actually what happens if you share a consented genome now. So here it is inside Synapse. It was scraped over to OpenSNP, which is a wiki based in the EU. From OpenSNP, it was scraped to a wiki called SNPpedia, which tends to sort of proxy whether if something is on OpenSNP and it's felt to have gone through enough of a consent process. But I only went through one consent process to have my genome, my genotype federate across these. And I got back an automated report, right, not worried about whether or not they were returning incidental findings or not. And so the previous speakers are sort of focused on things inside the clinical system. The argument I want to make is these things are happening already outside the clinical system, completely outside the scope of these sorts of discussion. So I got this back as an email. It's different than what I had from 23andMe. The hypertension increase is a bad genetic thing. The not bald part is a good thing. But the point being, there's no context to this sort of report. It's an automated algorithmic report. It's not sitting and deciding whether or not to return incidental findings. It's saying, here's what's known about your genotype, whether or not it's an incidental finding, and we're going to give it all to you. On top of that, it got syndicated out from there to someone else who looked at it as a genealogist and sent me an unsolicited email telling me that my parents weren't related to each other. And so all in all, right, the syndication and the federation of my genotype shows me that, first of all, a lot of these discussions that we're having today are happening inside an infrastructure that assumes it has relative control over the system, when in reality there's an entire separate infrastructure developing among consumers, among patients, among advocates, among passionate people, and among researchers who are simply going full force ahead. And although I was fortunate not to have any negative results that I didn't know about or to have any inbreeding reported to me, not everyone is going to be that fortunate. And the benefit, the reason that we want to do this despite all of this, is that putting together large numbers of genotypes and genomes and large numbers of phenotypic outcomes, whether it's health records or cell phones or what have you, has the potential to let us build really interesting predictive models of disease, of drug progression, of immunity. And so this is an article from the Scientist that shows how putting together genomic information, clinical information about breast cancer lets us build accurate classifiers that can, 3/4 of the time predict whether or not breast cancer is going to recur in a given population. So the goal here is three simple goals inside consent, which is, typically it's the relationship of a person and their clinician, and we wanted that to be an asymmetric thing where you have many people participating through a single consent process. That also implies you need to have scale and you need to have automation, or you won't be able to achieve that goal. So this is the consent process we actually put together. It takes the form of a wizard that walks people through an interface to the actual consent document. Because the consent documents have gotten sprawling, they've gotten out of control, we figured let's put a user interface around the consent document to call out what we think are the most important elements, and then we'll test on the back end and see if people actually understood what they were doing. So this is actually an IRB-approved study that's asking people to donate their data to research. We start with the wizard. You have the little crumb trail along the right. And what you see as you click through is you get very plain English text that says, listen, we're going to impose these conditions on researchers through terms of use. If you were to click on terms of use, it would pop up and say terms of use is a contract. We can't police it that much. Don't expect too much out of it, although we're going to do our best, and we're going to put some friction into the system. And we're going to ask people to commit to saying that they won't try to re-identify you even if they can. They won't hurt you even if they might have that power, and they'll share their research under open access terms. And there's a fair amount of evidence that making the researchers understand what they're doing creates a social contract that can be quite powerful. But we also want to make sure we're not promising anything to the user that we can't guarantee. Similarly, these are the rights you have to positively grant to researchers to do research, to redistribute data, to publish the results of the research, to commercialize products. You have to click on all four of these to move forward. At that point, you have to watch a video. The video is 7 minutes long. It's quite short compared to a clinical encounter. For a YouTube video, it's considered quite long. Most of the dropouts we've gotten in the process have happened during the video. We don't have fine-grained enough analytics to know exactly where or why inside that they did. They can mute it, they can walk away from their computer, but they cannot advance until the video has completed at least one play. And we focus on the potential for unanticipated harm, including incidental findings, but also including things like someone has your genotype and then they synthesize your DNA and place it at a crime scene. Because the incidental return of clinical finding is just one element of having genomes be out there in people's possession. And only at that point can you click and say, these are the three pieces of the consent that I want to provide. First, I understand the uncertainty and the risk. Second, I provide consent. And third, I understand that although I can withdraw, I can't withdraw data that's already been syndicated. Right? Because it's very hard to promise in an open context that you can delete all the copies of data, because data gets cloned, data gets backed up, and you have to bake that into what you're doing. And at this point, and only at this point, is the person confronted with the consent form for a digital signature. And I won't go through the rest since we're focusing on the consent part. But the early returns are that this implementation of web-based consent is effective at achieving at least our initial goals, which were to create a consent process that was easy to use and where people understood the core risks and benefits. We've had about 10,000 start the process. We've had about a 10% completion rate, and about 2 out of 3 people who have made it through have contributed some form of data, typically in the form of scans of their medical records, but in many cases genotypes, and in a few cases whole genomes. So in terms of the surveys that we ran, we have about 10% of those who have completed the process have filled out the survey. It's about 13%. And so you see we've had, typically we see the vast majority of people saying that it was either extremely or very easy, that it was extremely or very user-friendly, so we consider that really positive. This is an interesting slide, which is how much of the consent form did you actually read? And so only 31.7% said they actually read the entire form. This is consistent with what we expected to find, but that meant that the next slide was what we really wanted to be tested on, which is did you get the understanding and the comprehension? So do you choose what data to upload? We got 100% accuracy on that. The withdrawal, we got 98% plus accuracy. The idea that your data can be cleared from the system being a false concept, we were really happy to see that above 92%. We weren't quite as happy with number 10, the idea that you're guaranteed confidentiality. We got almost 59% people with what we consider to be the correct answer, but we're going back now and redesigning it to be even more clear that although we are de-identifying the information, we cannot guarantee any sort of confidentiality over the long term. And then this idea of assigning terms of use and the redistribution of data. We're really happy with the effectiveness of the consent from an informing perspective. What we need from SAGE, and I hope to have the conversation around today, is three things. First, we need incentives. Sharon, I saw, made a comment earlier this week about you can herd cats if you move food. I've heard it said that you can, if you want to herd cats, you just strap a bowl of cream to your back and run. And if we're going to have progress in consent, there have to be incentives for IRBs to participate in novel methodologies, for researchers and clinicians to participate, and for people to participate, so that it's not just me syndicating my genome and getting emails about inbreeding, but that I'm actually putting my data into the clinical system instead of putting my data outside the clinical system. And right now the incentives are not lined up to put together citizens, scientists, institutions, and the ethical systems that sort of guide science. So increasingly it's likely, if we don't get the incentives right, that a lot of citizens are simply going to stay away from the institutions, and I don't think that that's healthy. Second is we need network effects. We're starting to see open projects around genomes popping up, whether it's Personal Genomes Project, Reg for All at the Genetic Alliance, our own work at SAGE, but we need to start seeing these connect to each other. And if we don't figure out how to use consent in such a way that we can pull that off, the consents themselves, whether they return incidental findings or not, will segregate open projects from each other. Which leads to the last point, which is we need advice from ethicists and IRBs on how to make informed consent documents interoperate with each other. And not just the consent documents, the clinical protocols, so that we can begin to take the results of one study and use them as the inputs to new studies, so that we can take two studies and combine them with one another as the foundation for yet a third study. And that's not been how the governance structures around this have worked. The idea has been simply that we will segregate these information bases. But this sort of conversation we're having today is very important, but when it's cheap enough to sequence someone's genome for under $100, at that point it's cheaper than a blood panel. And so if there is a non-interoperable version of my genome that has been isolated because of incidental findings in a clinical context, but I can get that data generated as a consumer outside of that entire infrastructure, we're going to have some sort of system emerge. And what I'd like to make sure is that those two systems can talk to each other. And I'll stop with that and then look forward to the conversation. And feel free to email me if you have any questions or would like to see our documents. Thanks.
Fabulous. Thank you all. This was really a terrific overview and a deep dive. Lots and lots of richness here. I think people are probably going to want to watch this webinar to get all the nuances. And I'm sorry we couldn't actually give each of you an hour, because that would be the better way to go. We do have a number of questions. We're not going to get to all of them, so I'm going to throw out a couple of them. I'm trying to consolidate them, so if you don't hear your question in the questions that I'm presenting, we will try to get those answered later for you. So for the first one, and I think that all three of you certainly could comment on this, is it true that the current consent process is broken, or is it that the basic construct no longer fits the new state of the world? So if each of you could comment on that. That's a general one that actually encompasses several others as well. And make your comments short, and maybe you can just go in the same order, Amy, Susan, and John, and be very brief.
So I wouldn't say that the current consent process is broken. I think informed consent is an incredibly important mechanism for appropriately informing individuals of and giving them options to have things done in any context, clinical research or consumer. I do think, as John really nicely pointed out, that there's an opportunity because I think we've recognized for a very long time sort of the challenges of informed consent and of obtaining informed consent. And I think there's a real opportunity with sort of advancements in technology to think about creative ways to engage people and to increase understanding and to make sure that they're informed to the level that they would like to be informed in making decisions about their clinical care, their research participation, and what they do in their own sort of personal sphere.
So it's Susan. I think I heard in the question, should we basically chuck consent? And the answer to that is no. I think consent remains absolutely crucial. In fact, as genomics is moving into the clinical sphere, educating patients about it, what's involved, getting their consent is a crucial part, I think, of earning their trust. Their trust in the technology, that it doesn't mean all sorts of information about themselves is just released into the atmosphere and they've lost control, that they can still make meaningful choices in terms of the analysis that's done. I really think that medical genomics is going to hit a brick wall unless people have that trust and are confident that a robust consent process with its imperfections, which we are trying to improve, is going to be used. They need to be able to consent or refuse.
Yeah, I would just follow on to that. You know, the point I was trying to make was how the absence of consent in the emerging consumer system leaves me unprotected to find out things that I really might not want to know. And the key to consent, to me, is this idea of a balance between the risk and the benefit to participation in something. And what I think has happened is that we've gotten a little unbalanced in trying so hard to prevent any imaginable risks that we have foreclosed a lot of potential benefits by, and consent is sort of, you know, the innovation inside consent has stalled a little bit on that. But that's not a reason to chuck consent, right? It's a reason to rethink the way that we design consent, to bring some of the principles of user experience and user interface to consent, and to recognize that the scale at which people can have access to their data is changing, and that means the risks and the benefits are changing. But this idea that we should be willing participants, that we should have agency, that's a beautiful idea, and I don't think it's one we should get rid of.
Great, fabulous, thank you. So in the 3 minutes remaining, I'm going to ask one more question, and I'm consolidating several, so I'm doing injustice to the questions and also asking you to answer briefly, which will do injustice to your answer. And the question is a really complicated one and actually asked by several people from several directions, and that is rights. So do individuals have the right to incidental findings, even ones that have not been deemed actionable by a guideline system or a medical system? And at the same time, the issue for clinics and others to find the time to do the right informed consent, to understand that. And then for the physicians, how do they understand kind of the unrolling iterative nature of what's actionable and what isn't? So in other words, I think what we're looking at is the relationship between the physician and the patient in a system that I think each of you have said very clearly is quite iterative and quite emerging. What do we do about the rights of whom in that structure? And I think we probably can address both legal rights as well as sort of ethical rights, and you can go wherever you want with that question.
So this is Amy. I'll start. I think the point that Susan really articulated very nicely in her presentation is critical here, which is that rights and obligations derive out of, they're role specific, they derive out of relationships. So an individual's right in a consumer context is very different from what rights they have in a medical context where there's additional constraints and such imposed by the system, and it's also very different from the rights that they have in a research context. So it's very difficult to answer sort of what are the rights sort of in a vacuum, because it very much depends on the context that you're in and the relationship with the individual who you're claiming a right against. So a right has a corollary obligation, so what obligation do they have to you? And so I think that the more we kind of, to use Susan's articulate term, mush together these realms of research, clinic, and consumer, the more difficult it is to talk about and sort of tease apart the rights and obligations of different individuals.
So I very much agree with Amy, but let me take it a step further. I love this question because it captures a real problem that we have taken a very clinician-centric view of what's actionable, and, you know, we, the professionals, the experts, are in charge of what are the criteria for offering back these incidental findings, some of which are extremely important, maybe even life and death. And I think, if I had to predict, there is going to be a real pushback against that. There is going to be an articulation that these are ours, the participants, the patients. And particularly in the clinical sphere, the notion of what the physician is obligated to share with the patient is not, as I suggested in my presentation, limited to what the physician thinks she or he can fix. So there is pushback coming. It's necessary. Actionability should not be defined solely by what the clinician feels she can fix. It really should be more broadly defined by what research participants and patients find material and important in those two different contexts.
And John, do you want to comment?
I think that Susan and Amy have done it wonderful justice. So the only point that I would raise is just how rapidly what we think is actionable changes, and how many different sources there are to create that change, whether it's computational models, whether it's new publications, which are increasing in volume and frequency, or whether it's new machines that let us, find out. Because it's very probable that we might return raw data to people that we feel is not actionable that turns out to be actionable 6 months, 9 months, 12 months from now. And so it is such a moving target in terms of our ability to understand the genome that coming up with definitive statements on what's actionable and what isn't are basically guaranteed to be out of date by the time they achieve consensus.
Great, thank you. And as we know, by all of medicine, that seems to be true as we learn about ourselves and our health and disease and prevention. I want to thank the three speakers. Your presentations today were fabulous. They really deeply covered the topic. I am sure that the community really appreciates this for the whole, and the series itself is really quite enhanced by what you offered, so thank you very much. I apologize to people that we didn't get to your questions, and if any of them are very specific, I will send them to the speakers and we'll get the answers. I remind everyone that our next webinar in this series is on recommendation 4, which is on July 9th, and our speakers are Paul Billings, Stephen Friend, and Laura Rodriguez. I also invite people to our webinar series on Registries for All, which addresses some of these same issues, and that's a whole separate other series running. You can see it if you go to the front page of Genetic Alliance and click events. You can see those webinars as well. And thank you to all the participants for putting such great questions into the portal. I can tell that this is a topic that really jazzes the community and that we all have a lot more work to do on, and I am encouraged by your work, the three panelists, as well as those who are participating.